Our next guest is Assistant Professor Alison Shield. Now, this is very interesting because Alison's PhD student Sue Mahat applied for a grant from the CMT Association, which he received, of $4,000. And that grant was specifically for the purchase of a standard dual-controlled vertical electrophoresis system, or something along that line. Anyway, pardon my pronunciation. And to tell us how the grant is being used and what's been happening, here's Alison. Thank you.
Thank you. Sue hasn't been well the last couple of months, so I'm stepping in for her. And I wanted to give you a little bit of background on what we've been doing, and actually, very coincidentally, the last question leads really well into some of the stuff that we're doing. So indulge me in a little bit of a mystical, magical tour. I'll try and stay on track. The nice thing about the audience that I have here is I don't have to do my usual background about what CMT is. Hooray! So we can cut some time off. This is a picture of Sue, and I wanted to just start off by talking a little bit about her because she's not here and can't present. Sue comes from Nepal, and she actually has had an interest in clinical sciences for quite a while. So she originally started off doing a master's in microbiology, looking at pregnant women and looking at infections in pregnant women. Then she came to Australia and did another master's degree in biopharmaceutical research, and finally decided that research was really her thing, and she started a PhD. And she would be far too modest to admit this if she was here herself, but she has personal experience with neuropathy. She has a transverse myelitis that impacts on her lower thoracic and has some sensory loss and some movement issues in her legs as well. And she'd never admit that, but I know that that's why she's interested in doing research in this kind of area. Oops. So GDAP1 is a pretty funny place to start, but it's the protein that we're interested in. Why is it called GDAP1? For a completely random reason. There was a paper back in the late 90s by a Chinese group who were looking at neural development in mouse cells. And they found some proteins that had changes in expression levels, and they happened to call them all ganglioside differentiation-induced proteins, because they did. So we've been stuck with this name GDAP1, but really we don't know what this protein does. What we do know is that it's linked to Charcot-Marie-Tooth, and I wouldn't be here if it wasn't. It's a fairly rare form of Charcot. It's in most cases, a severe recessive form that's picked up in children when they start to walk. To my knowledge, there aren't any cases of people with mutations in this particular gene in Australia. That doesn't mean they're not out there, we just haven't necessarily found them yet. Over the last 10 years in the research that's been done, and this is exactly the same table that Garth just showed you, what we've found is that GDAP1 is associated with many different forms of Charcot-Marie-Tooth. So primarily it was the CMT4A variety, but it's been found in families that have CMT1 and CMT2 as well. And that's quite interesting, because often we have one gene that's associated with one type of CMT, and here we have a gene that seems to be crossing over those boundaries and causing physiologic effects in nerve conduction and other things that really is quite diverse. And we also know that sometimes people that have this mutation have really severe forms and sometimes they don't. Sometimes they have vocal cord involvement and other sort of symptoms. So here's an example of a gene that seems to be causing a spectrum of disease, and this is quite interesting to us. The reason why I've highlighted MFN2 is MFN2 is another one of these fairly common genes, and it's involved particularly in CMT2 at around 3% of people. I've highlighted it because both of these genes seem to be involved in changing the way that mitochondria work within cells. And so this sort of started us thinking, well, maybe we've got a common pathway, and if we can understand how this pathway works in normal people, then maybe we can translate some of that information into people that have disease. So we're actually not so interested in looking at that rare family that has the mutations in the gene. That'd be great if we had it. But what we're really interested in is saying, well, how is this gene potentially involved in a pathway that could then be contributing to modifying factors or other factors involved in disease? but we don't know what it does. And the way that I came at this was quite by accident. I haven't come from a clinical background. I haven't had previous interest in peripheral neuropathies. I've come from a background where I'm really interested in how we metabolise drugs in our body. And we have some enzymes, suites of enzymes that are found in our liver that are involved in making sure that anything that we take in our mouth, whether it's a medicinal product like paracetamol or whether it's a recreational drug or anything else, how that passes through our body and leaves the body safely. When we started having a look at this interesting protein, it looked like it was one of these glutathione transferases or GSTs. And we can do all sorts of structural modelling in the lab to have a look and try and predict, well, what does this protein look like? What might be its function? So as I mentioned, GSTs are metabolic enzymes found in the liver. They're found in everything from bacteria through to humans. They're able to conjugate a chemical reaction with an antioxidant molecule called glutathione or GSH. They don't just metabolise drugs, they also look after oxidative stress in cells. And oxidative stress is a normal process. We breathe oxygen in, our oxygen is able to be processed by our mitochondria. As part of that processing, it produces energy, but it can also produce these species called reactive oxygen species that can cause damage. Our bodies have ways of getting rid of that. And that's where some of these ascorbic acid trials and curcumin trials and other things start to fit in as well. This last bit's a little bit cryptic at the moment, but most of these GSTs are found in the soluble part of cells. So when we mush up cells, we have bits that are able to be very easily dissolved in water, and we have bits that are not. And so the soluble bits are the bits that like to be in water. Now, GDAP1 is a little bit unique. It structurally looks like it might be a GST, but it doesn't seem to have those functions. It doesn't seem to do these conjugation reactions. It doesn't seem to bind onto glutathione, not that we can see. And actually, it's found in the mitochondria, which is once again unique because most of these mitochondria are non-soluble compared to the soluble fractions of cells. And this pretty colour picture here that you might not quite have good colour contrast on, we've done some labelling studies here, and this is just a normal cell culture model. It's not from CMT or anything else. This light green that you can see is showing the soluble parts of the cell. The red is showing you the mitochondria. And when we look at where our GDAP1 is located, it's not located in this soluble part of the cell. It's in fact located right around the edge of the mitochondria. So this gives us a clue that it has something to do with mitochondrial function. Why are mitochondria so important? The hypothesis came out that GDAP1 might be a regulator of mitochondrial networks. Mitochondria are really important in nerves because nerves need lots of energy. What are nerves doing? They're transmitting information from one part of the body to the next part of the body. They're long and they require that energy to travel through that cell in order to then have transmission. So what happens in mitochondria is we have these networks and we have this balance between these processes called fission and fusion. So where we have fused mitochondria, they're very long mitochondria that can transmit energy, but we have fission processes where we can break up those mitochondria and get rid of damaged mitochondria and try to make sure that that system's healthy. Now this is a little bit of a complicated picture. Maybe I'll show you the simple one first. This is a cell once again. It's not a nerve cell, it's just a cell culture model. And you can see that this shows a very nice network of mitochondria where they're stretched out throughout the cell. This cell up here, once again, has a big shape. You can't see the whole shape of the cell, but the mitochondria are very collapsed in that cell. So one of them has good fusion, the other one is much more fragmented. And these processes are controlled by a whole host of different proteins. MFN is one of them, and once again we know that MFN2 is involved in CMT disease process. And it's involved in these fusion processes. And GDAP1 seems to be involved in these fission processes. So there seems to be this balance that maybe if we can understand how this balance works, maybe we can manipulate. This just shows you a time lapse of GDAP1. And remember the last picture I showed you, we had GDAP1 labelled in green. So what we're seeing here is very low levels of GDAP1, and this has been manipulated in an experimental model. We have increasing amounts of GDAP1 being expressed, which you can see as the green becomes more intense. And what you can see very clearly is our nice, extended, fused mitochondrial network, as we express more GDAP1, is becoming more and more fragmented. So once again, it's obviously involved in this process. We don't understand how it's involved in the process. We can see the process, we don't understand the process. The other really interesting thing that sort of came out fairly recently, and once again this is an international group, is that we're starting to see families that have combinations of not just one mutation that's causing disease, but several mutations that's causing disease. That's very interesting, and it's very interesting to see that this poor individual family has really screwed up mitochondria. We can assume that because we're not going to be able to do fission reactions and we're not going to be able to do fusion reactions. So this is probably the underlying reason this family has disease. We know that these things are balancing each other out. We don't really understand why. And as Garth mentioned very nicely in his talk, not only do we have these disease mutations, but in every single individual in the world, we have natural variations in our genome. And so I can guarantee that in our room, if we have some people that have MFN2 mutations, and if we don't, that all of you will have some form of variation in your GDAP1. Now, is that contributing to your disease? Maybe, maybe not. We don't really know. But it gives us a starting point to start thinking about some of these modifier genes. And the biggest problem with looking at modifier genes, and we found this particularly in cancer models, is it's really hard to find those effects. Now, Garth's been talking about getting down to less than 1% for a particular gene and 42 genes and how much money it costs and everything else. It's really expensive to look for just the disease genes. Then if you have something that's got a 0.01% modifier effect, you're starting to look at huge cohorts of families. And in fact, if you start looking at breast cancer models, for example, the studies that are needed are studies that have thousands and thousands and thousands of individuals. And those individuals all have one kind of breast cancer. So imagine if we did choose CMT1A, how many people would still need to then find those modifier genes. It's lots, and it starts to become limiting with the size of our population. And even when you start going to international cohorts, it can still start to be limiting. But we think GDAP1 is a good target to start poking at, so that's what we're doing. The crazy piece of apparatus that Daryl very eloquently tried to introduce has been very kindly provided by the foundation. Sue, off her own back, about a year ago decided that she wanted this piece of equipment so she could do some experiments, and I said, budgets, did my supervisory role, and she said, well, hey, I found this foundation, do you think I should write to them and see if I can get some money? I said, it can't hurt, see what happens. And so she put together a proposal, and working with the executive, she was able to be awarded a small amount of funds to get this piece of equipment. Now, it's not this actual one, but what I wanted to show you is what it looks like and what it actually does. So you'll remember I talked about soluble forms of protein and non-soluble forms of protein coming out of cells. The non-soluble forms of protein are really hard to investigate because these techniques rely on soluble proteins or ways of solubilising proteins. We can solubilise proteins by using detergents, but often the detergents we use in the laboratory are quite aggressive and will break down the structure of a protein. So if you have two proteins that are stuck together, then you add protein to them, add detergent to them, they'll fall apart. And you won't know if they were ever joined together or not. What this particular apparatus allows us to do is to mix in a different kind of protein, a different type of detergent called Chimassi Blue, and run those complexes together on a gel. And normally we would use these for just regular protein investigations. Once we've contaminated a set of equipment with this detergent, we can't then use it for normal gels. So we need to have a dedicated piece of equipment. We can run it with this beautiful blue coloured dye. It solubilises up those complexes, and we end up with rows of bands that then represent not just one protein, but proteins working together. And this is what it sort of looks like at the other end. Normally, the protein that we'd be looking at would be of a size about 37 kilodaltons, and it doesn't really matter what that size is exactly. But when it starts to run as a complex, more proteins, we'd expect it to be much bigger. What you can see on this bottom gel here is that we have things that are more like 400 kilodaltons. And this is starting to tell us that this doesn't just contain GDAP1, but it contains GDAP1 associated with some other proteins. And so now what we can do is we can go in and we can characterise these bands and we can figure out what else GDAP1 is playing with. By understanding what the proteins are in that complex, we can start to put together a picture of what the pathway is and how it's involved with each other. So we can look at this and say, oh, well, is MFN2 actually involved in this pathway as a complex? Or is FIS involved? Or is it something else? And whatever it is, how does it work? And therefore, is it a target that we can potentially manipulate? So we know that changes in mitochondrial morphology lead to disease, and we know that they look different when we look at different patients. That's easy. We look at patients, we look at their genotype, we put the two pieces together. We're speculating that changes in GDAP1 are not just causing those disease phenotypes when we have a mutation, but some of those subtle differences in natural variation of GDAP1 might be modifying effect in disease carriers. This is, as I said, speculation is put up there entirely on purpose. It is speculative. We need the experimental data to show that. Once we have experimental data to show that, then we can start thinking about, well, can we use this information to make new approaches to try and help people to decrease the severity of their disease? How soon is that? That's always the question people ask. We don't know, because we don't really know where the research is going to drive us to. But we hope, like we always hope with these sorts of studies, that we can find some little key that can help in some small way. So finally, thank you again to the Foundation for providing the funds to buy the equipment. Sue is raring to get back into the lab when she's well enough. As with any research program, there are a large number of individuals in different places that have helped over the years to try and develop the program. Once again, here's Sue and me, and we also have Khulud and Cindy who are working on other aspects of the project and trying to understand what normal variation is in this particular enzyme family. So thank you for your time, and I think probably Daryl needs to move on with the next speaker, but I'll be happy to answer questions this afternoon in the panel session.
Thank you very much.